Hello, everyone, and welcome to today's webinar, What's in Your Sample? How Sample Biology Can Influence Fluorochrome Choice. I am Kaylee Bach of Labroots, and I'll be your moderator for today's event. Today's educational web seminar is presented by Labroots and brought to you by BD Biosciences. To learn more, please visit bdbiosciences.com. We encourage you to participate today by submitting any questions you may have during the presentation. To do so, simply type them into the Ask a Question box and click Send. We'll answer as many questions as we have time for at the end of the presentation. You may also submit any technical issues here as well if you have trouble seeing or hearing the presentation. And with that, I'd like to now welcome our speaker, Kim Lee, Clinical Application Scientist at BD. For a complete biography on our speaker, please click on the Presenter tab. Kim, welcome. You may now begin your presentation.
All right, thank you for that introduction. Hi everyone, my name is Kim Lee. I am a clinical application scientist with BD. Thank you for joining me today. I am very excited to kick off the panel design three-part series with this webinar, which focuses on how sample biology can influence fluorochrome choice in designing flow cytometric panels. The second talk will cover the characteristics like fluorescent spillover and spread that occur when you include multiple fluorochromes in a single panel and how to mitigate those effects. And once you have taken the sample biology and fluorochrome considerations into account, the third talk will cover how to standardize your assay for reproducibility and how to troubleshoot existing panels in your lab. So please make sure to register on three webinars in this panel design educational series. To begin my talk, first, let's start with a question. What is the goal of multicolor flow cytometry? It might be taking a bunch of cells in a sample and divide them into distinct populations by using fluorescently labeled antibody. So we can study things like how prevalent they are in that biological sample and how they look different than what is normal for cells. For example, are they increasing or decreasing the expression of certain markers for normal? But how do you judge when something is increased or decreased? It is when you can gauge on the population of interest to characterize it. And when you can recognize a change in expression, sometimes a subtle change in expression from brighter to dimmer can make a big difference in overall immunophenotypes of cells of interest. So let's get back to our question. What is the goal of multicolor flow cytometry? It is the ability to gauge on the cells of interest. I would like to pause a little here to walk down the memory lane so that we can all appreciate how far the technology for flow cytometry has grown, and BD is there every step of the way and plays a vital role in this growth. BD has been on this flow cytometry journey for a long time, more than 45 years to be exact. And as you can see, BD has many firsts in this journey. BD had the first commercialized fluorescent activated cell sorter on the market, and this is where the FACS moniker began. And in 2017, BD launched the FACS Lyric, which is the first clinical analyzer that has up to 12 colors. And then 2 years ago, with the FACS Symphony, that can do both conventional and spectral flow cytometry. And to present day, where we now just have launched the first cell sorter called the FACS Discover that allows you to look at images of the cells you want to sort. But BD innovation does not stop with instruments. As instrument evolved, so has the number of fluorochromes. We have spent a lot of time studying this evolution, and the lessons we learn are the one you will hear about in this educational series. We have a lot of choices when it comes to fluorochromes. And our toolkit to answer that basic question in flow cytometry, what in your sample, has really expanded for doing multicolor flow cytometry. So think about what panel you are using in the lab, whether they have 6, 8, or 10 colors. Have you thought about adding more color to your panels? And in doing so, what are the advantages? So it could be fewer samples to prepare, which is important when you have limited amount of sample. Oftentimes in the lab, you need to split a hard to collect sample for many ancillary tests. By the time it gets to flow cytometry to your lab, there is a tiny portion, and you need to make use of what you have. Or another advantage is could be shorter time to results because you would have less tube to analyze, or it could be having fewer redundant markers in each tube, and that in turn saves money. Or it could be more comprehensive information about sample. The answer is all of these are the advantages of adding more color to your panels. For example, you might have started testing T-cell enumeration using 3 colors, CD45, CD3, and CD8. And that is grew over time to also include the counts and the absolute count and the percentage for CD4, T cell, NK cell, and B cells. So your panel has become larger and larger to accommodate the need for more information. But with more colors come more challenges. This is why throughout the educational series, we will focus on the 3 basic pillars for panel design: cell biology, fluorochromes, and cytometer. The first pillar is know the biology, which include antigen density and antigen co-expression. This will be the topic of my talk. I will discuss the expression levels and patterns of the markers on the cells. The second pillar is know the fluorochrome. It involves fluorochrome properties such as the brightness level and resolution, and how different fluorochromes react with each other in a multicolor panel, and result in spillover and spread. You will learn the tactic to mitigate the spillover and spread in different channels. The third pillar is know the instrument. You will learn about instrument-related factors such as configuration and sensitivity that can influence panel design for standardized and reproducible results. Ultimately, you will learn how to use the 3 panel design fundamentals to troubleshoot your own panels. Again, our goal for a flow cytometry assay is to get distinct cell population so we can gauge them. In this case, we do gauge on 2 populations that are relatively easy to gauge because one cell population express that marker and the other one doesn't. But what did you do when the positive population is dim? Like for example, like here, what do I even mean when I say dim? How do we measure this dimness or brightness? How do we measure the resolution? So there is a metric for this. You may have heard of the term stand index. It's the ratio between the brightness and the width of the negative. As you can see, these 2 components impact the stand index and or fluorochrome resolution. let's talk about the brightness. Brightness is simply the difference between the MFI of the positive and the MFI of the negative. Brightness is a function of reagent in your assay, antigen density, and fluorochrome yield. So it can also depend on the instrument settings such as PMT voltages. How about the width of the negative population? Is it measured by the standard deviation of the negative population, which you can find from the statistic table from any analysis software? The width of the negative is influenced by instrument intrinsic factor that you can monitor with your daily QC and spillover and spread. Some of these will be covered in other part of the webinar series. Maybe we think about this in practicality. Imagine how antigen density specifically might change the standard index of this plot. For example, if this is CD8, it is highly expressed on CD8 positive T cells. And there is a nice separation between CD8 positive and CD8 negative cells. But what if you are looking at another marker, not as bright? How might an antigen with lower antigen density affect the resolution? So as with the example of CD8, resolving the bright population is very easy. But reality is not as rosy. You often have to deal with antigen with lower antigen density resulting dim expression. So let's look at some scenario where things have an impact on resolution of a dim population. Notice that the MFI values of the bright and the dim population are the same in the three sample, but the standard index might change depending on the negative population. The top example show that the negative population has low background, and you can, in this case, confidently gauge out the dim population. So this is the ideal situation. But what happens when the negative population has high background, and that results in poor separation between the negative and the negative and the dim population? An example I can think of is the expression of CD117 on maturing myeloid cells. Maturing myeloid cells in bone marrow have high background and express a range of CD117 maturation. The standard index or overall brightness had changed because the negative population has, with high background, have shifted to the right. So as you can see, the dim population is not well resolved. Similarly, the resolution is not good when you have a negative population with low background, but high spread. So what is spread? Spread is a phenomenon introduced by spillover. We will discuss about spread in the next webinar, but you can clearly see that the effect of spread in resolution, the separation between the negative and the positive population is diminished. So in the second and third case, even though the bright population is really easy to gauge on, the main focus is the ability for us to gauge on the dim population. So far, I've given you the background and example of how knowing the biology is important in panel design. Let's now explore more about the first pillar of the panel design fundamentals. Specifically, why is it crucial to figure out the antigen density and the antigen co-expression? So to design a panel, you typically come up with a wish list. In this wish list, you need to take into consideration of the type of sample you're testing, which population of interest you might find in that sample, and which markers you can that can help you to achieve that goal. So using that wish list, you begin with creating a population tree like this, starting with the most generic lineage marker like CD3, then get more and more specific. From there, it branches out to CD4 positive and CD8 positive population, and there are subsequent subsets. And then you can decide which one are your critical population. It is then important to find out what antigen or markers are expressed in this population and at what levels. It is also important to note that which marker are co-expressed on the subsets of interest. In our example here, we are looking at subset 1 or Treg, and subset number 3 or CD4 memory T cells from the CD4 positive T cells. These two subsets both express CD3 and CD4, but there are a couple differences between them. Subset 1 expresses CD25, whereas subset 3 does not. The other difference is the intensity of CD127. Subset 3 expresses brighter CD127. So knowing these features, aka the biology, will help you to validate that, yes, the marker you in your wish list will help you in identifying the population of interest, or in a simpler term, knowing the biology will help you in the panel design process. It is also important to understand that the expression can change over time, or with certain conditions such as after treatment or reactive. For example, what we have here within the same subject here, marker density can vary slightly. So once you know which antigen you are using, you can then classify them based on expression. Primary antigen are expressed at high level, and cells are often either positive or negative for these markers. So these are the easiest one to get on. Primary antigen typically define broad subsets or lineages. Example of primary antigen are CD4 and CD8, and they are highly expressed and expressed in an on and off fashion, which means having a clear negative and a clear positive population. Next, there are secondary antigen. These are also well characterized, highly expressed, but usually over a continuum. And there are not clear positives or negatives. An example of this would be CD38, where it is negative to dimly expressed in mature B cells, but relatively bright for hematochrome and very bright for plasma cells. Tertiary antigen typically expressed at lower levels, at low level, where their expression level may change in normal or disease state, or they are, unknown marker that you are not sure how the cell might express. Your pathologist might come in and say, okay, I want you to test, to bring on the BALL MRD panel, and in his or her wish list, there may be like CD58, and you never worked with CD58 before, and you don't know how that, the cell would, express with CD58. So let's talk about CD58. CD58 expression changes in hematogones and B lymphoblastic cells. So basically, it changes in a normal condition and in the disease state. Hematogones or normal immature B cell express moderate CD58, whereas B lymphoblastic cell often express bright CD58. So in this case, a pairing of CD58 with a right fluorochrome will help you to recognize the subtle changes in antigen expression, and in turn make the right diagnosis. You could tell if the patient has, residual BALL cells in the bone marrow after treatment. So to summarize, sample biology can influence panel designs. Antigen density is a function of the biology of the cells. It is dependent on the sample, and antigen density along with the fluorochrome brightness can impact resolution. And last but not least, classifying antigen based on level of expression help you with the fluorochrome pairing process. So this brings me to the next part of the talk. Know the fluorochromes. As you recall, this is the second pillar of the fundamentals of the panel design. So to illustrate the importance of fluorochrome pairing, we have an example here of CD3 versus CD197. If you don't know what cell express CD197, it's okay. We just want to show how the right fluorochrome can reveal biology. So back to our dot plot here. They were from the same sample. CD3 on the y-axis was paired with the same fluorochrome, whereas CD197 was paired with different fluorochromes. The fluorochrome from the bottom row are considered dim. If you want to gauge on the positive population or on the double positive population, you might be able to do so with PerCP Site 5.5, but it's become harder here with FITC. You don't see a clear separation to make a distinction where the double positive population should be. But if you look at the top row, where the CD197 was paired with the bright fluorochrome, you don't have any problem gauging on that double positive population, from PE to Alexafluor 647. But what you also notice is from the top row is that there are 4 distinct populations, especially here. The CD3 negative, CD197 positive population is clearly shown on display, whereas you couldn't see that population in the bottom row where the dim fluorochrome was used. So this drives home a point of why knowing antigen density, where in our example here, CD197 is a low antigen density marker, can really influence whether you should pair it with a bright or with a dim fluorochrome to effectively gauge on the positive population. So let's learn a little more about fluorochrome. All instruments come with the blue laser. So you are probably already familiar with FITC, PE, and PerCP. These, so they are organic and protein dyes. In this example, FITC is an organic dye, and PE and PerCP are protein dye. Then there are tandem dyes. The tandem dye were originally invented to expand the menu from the same laser line. In our example here, PE size 7 is a tandem dye. So with this, you have the option of using 4 instead of 3 different fluorochrome from the blue laser. Some other lasers even have more choices of tandem dyes. So what is tandem dye? Tandem dye is a combination of 2 dyes. One dye, in this case, PE, acts as an energy donor. So when this PE molecule is excited by the blue laser, this will transfer energy or electron to the size 7 molecules. The size 7 dye molecule will get excited by this energy and emit light at a larger wavelength. This phenomenon called FRET, fluorescent resonant energy transfer, In our example here of PE size 7 here, the emission wavelength for size 7 molecule is 778 nanometer. But there is also a residual amount of emission at the lower wavelength detected in the PE channel. So there are So knowing this, so there are a few considerations when handling tandem dyes. And this brings me to the next questions. How do tandem dyes need to be handled? Do they need to be protected from light? Do they need to be protected from high temperatures? Do they need label and lot specific compensation control when you are using them in a multicolor flow cytometry panel? Or is it all of the above? The correct answer is all of the above. As you know, tandem dyes, you have like two different molecules connected together. So it makes sense that, these two are more, the tandem dyes are more fragile. So, so you need to, more prone to break down. So you need to protect it from light. You need to protect that from the high temperatures. But let's talk about label and lot specific compensation control. So tandem dyes are special and they do need label and lot specific compensation. So in this example, we have three cocktails with three different PE size 7 conjugate, CD8, CD4, and CD3. In the example here, we are looking at using CD4 PE size 7 as a compensation control for all cocktails. So this happens quite often in the lab because it's more work to create more three different compensation control with three different conjugate. And we think, okay, well, potato, all PE size 7 markers are the same. But what happened in the lab dot plot here with CD8 conjugate is that because CD8 positive T cell have higher antigen density than the CD4 T cell. So this sample end up being, undercompensated. In the middle plot, because we use CD4 as a compensation control, this sample is properly compensated. On the right, however, CD3 cells have lower antigen density than CD4 positive T cell. So this sample is overcompensated. So when we use label specific control, bada bing, bada boom, Bob's your uncle, all the sample are properly compensated. So what is the message here with tandem dyes? The message here is we should take our time and create label and lot specific compensation for each of the sample, even though the task might appear tedious and time consuming, because really your result is more important, patient care is more important. You want to get a right result. So your instrument might have a red laser in addition to the blue laser. And here are some fluorochrome excited by the red laser, APC R700, and APC S7. But are any of these tandem dyes? Here, APC R700 and APC S7 are tandem dyes of APC. You can guess these are tandem dye because of their names. Oftentimes, tandem dye have both donor and acceptor molecules in the name, but that's not always the case, and I will show that later what I mean by this. So modern instrument can also come with a violet laser. Violet laser is great because it can yield Syrogen dyes. So what are Syrogen dyes? Syrogen dyes are conjugated polymers. They have multiple optical segments that get excited and then get large collective optical response, resulting in a very bright signal. their brightness level is equivalent to or even brighter than PE. And similarly, tandem serovision dye are also very bright. So here you can see 4 different serovision dyes from the violet laser, BV 421, 510, 605, and 786. But we are not just limited to these dyes. There are others that you can use as well for different channel in the violet laser. But from this list, any of them are tandem dyes? From the name, you don't see the typical format where both donor and acceptor molecules appear in the name like you saw previously with PE size 7, ABCR 700, or ABCS7. But looking closely at the spectrum here for BV605 and BV 786, you can see the residual fluorescence in the BV421 channel from both of these fluorochrome, BV605 and BV786. So yes, they are indeed tandem dyes, but their names are not intuitively named, and you don't see that a typical donor and acceptor in the name. And in this case, BV421 is the donor of BV605 and BV786. So that brings me to the next question. If you use serovision dyes, how do they need to be handled? Do they require label and lots, specific compensation? Do you need to add BD horizon, brilliant stand buffer, or both? So as you can see previously, they have a common core. So yes, label and lots, specific compensation is required. But how about brilliant stand buffer? What is it? It is a buffer used when preparing sample when more than one serovision dye is used. It's reduced background noise caused by dye interaction. Let's look at our example here. One thing I like to emphasize is that these cells are compensated correctly because we check the single stand control. But as you can see in the top dot plot where no brilliant stand buffer was used, the cells appear to be undercompensated. But like as I mentioned before, these are correctly compensated. But when you add in the brilliant stand buffer, and you can see in the bottom row, the artifact or the higher background go away. So it is important to note that when you deal with more than two or above brilliant stand buffer, BV dyes or serovision dye, that you need to add the brilliant stand buffer into your sample, and same go with your control, compensation control as well, including the COMBIS particle. So to recap, we talked about the antigen density and how that might affect the brightness position of the stand index. But fluorochrome choice or fluorochrome pairing might also impact the brightness. We discussed about how important it is to pair the correct antigen density with the correct fluorochrome to reveal or identify the cell of interest. So here is another example. We've taken the same marker in the x-axis and combine it with different fluorochromes. The fluorochrome brightness is ranging from dim on the left to bright on the right. In this case, a secondary antigen is used. As you recall, secondary antigen are highly expressed over a continuum, and there are not clear positive or negatives. So to be able to see the dim or intermediately expressing population, we need to use brighter dyes. As with our example here, the dim population is not seen with FISC or APCS7, but we can clearly see with brighter fluorochrome such as BV421. BV421, is one of our brightest dye in the market out there. So what is the message here? Fluorochrome choice ultimately depends on the biology of your population of interest, and the dim fluorochrome would be appropriate for resolution of highly expressed marker like those primary antigen, and a brighter fluorochrome would be required for resolution of dim of intermediately expressed markers. So let's talk more about fluorochrome. Fluorochrome resolution can vary from instrument to instrument. And so you need to figure out empirically how each fluorochrome performs on a given instrument based on configuration, sensitivity, or instrument setup, etc. In this case, we evaluated how varying fluorochrome perform on a 3-laser instrument using CD4 reagent. Once we have the result, we start ranking and grouping them in brightness categories, so that's very bright, moderate, and dim. But realistically, do you have time and resource to do this for all instrument in your lab? Of course not. So BD has done this for you. We have taken out commonly used dyes and ranked them by their relative brightness. As you can see here in our table, I'm just going to mention a few commonly, the fluorochrome that you know, like PE and APC are bright fluorochromes. FITC are moderately bright, and some dim fluorochrome are V450 and APC S7. So what you will notice is that brilliant violet are some of the brightest dye out there. Most are as bright, if not brighter, than PE and FITC. So why is that important? Because it gives you the flexibility in pairing fluorochrome and antigen. So going back to our Know the Biology guide here, where we have 3 different groups of antigen. Primary, secondary, and tertiary. So how do we pair them with fluorochrome based on their brightness? You can pair your primary antigen with dim fluorochrome, secondary antigen with at least bright or moderate fluorochrome, and tertiary antigen with very bright fluorochromes. So this brings me back to the previous point I made about the BV dyes having different level of brightness. They can be paired with different classes of antigen so that you have the flexibility of incorporating these dyes into your panels. And when you have more channel on your instrument than your reagent in your panel, you can use brighter dyes when possible to ensure the panel is optimally resolved. So let's put this into practice. In this case, we have a set of dot plot of CD3 on the y-axis, pairing with 3 different markers, CD4, CD27, and CD25 on the x-axis. So CD4, CD27, and CD25 represent 3 different level of antigen density, ranking from high for CD4 to low for CD25. And as we learned previously, antigen density dictates which fluorochrome to use. So in the first column, we are looking at CD4 in 3 different formats. APCS7, which is a dim fluorochrome, per CP Sci 5.5 is a moderate fluorochrome, and BV421 is a very bright fluorochrome. For something like primary antigen, where they have high antigen density, like CD4 in this example, we would be able to gauge on the double positive population regardless of the fluorochrome choice. So if you look at down here, even though it's dim, but you still can gauge it on S, and then with the up here, it's really easy to gauge on the double positive population. But let's move to the second column, where we look at CD25 CD27, which has a moderate antigen density. And we are interested in gating on the double positive population. We can see that there is no real double positive population when we pair CD27 with a dim fluorochrome like APCS7. That double positive population become more resolved with per-CP Cy5.5. And when we look at the bright fluorochrome like BV421, not only we can see the double positive population, we could also see the single positive population that are clearly more resolved. So in this case, so we can see 4 different populations, whereas down here we can only see 2 populations. So, and the fluorochrome choice become more crucial when we look at the third column, where an antigen with low antigen density like CD25 here is used, with the moderate and dim fluorochromes like per-CP Cy5.5 and APCS7, we don't see the positive population at all. Only when we use a very bright fluorochrome like BV421, the double positive population start to emerge. So to put this all together, the rule of thumb in determining antigen fluorochrome combination is to use brighter fluorochromes for low expressing markers and to use dimmer fluorochrome for highly expressed markers. So that concludes our talk. So today we covered the benefits of multicolor flow cytometry panel. We described biology-related factors that impact panel resolution and highlight how to match antigen density with fluorochrome brightness. So please join us next time to go over some fluorochrome-related factors that can influence panel design like spillover and spread, and how to mitigate them. And the third and final talk, we will go over the instrument-related factors that you need to keep in mind to standardize your assay and how to troubleshoot the panels that are existing in your lab. Thank you, and I will take any question at this point.
All right, wonderful. Well, thank you so much, Kim, for your informative presentation. We will now start the live Q&A portion of the webinar. So if you have a question that you'd like to ask, please do so now. Just click on the Ask a Question box located on the far left of your screen, and we'll answer as many of your questions as we have time for. All right, so let's get started. Kim, I've got some questions already in the studio here. This first question asks, what type of antigen are intracellular or cytoplasmic markers?
Oh, that's a very good question. So those should be, you should consider that or treat that as tertiary markers because it can be hard to determine exact antigen density for these markers. Oftentimes, you don't deal with these markers in the daily basis. And some of them, like MPO or cytoplasmic CD3 can be the lineage marker. So you really, the make or break expression of that would, the expression of that would show, if the certain marker, certain cell would be like the has the like the T cells lineage or myeloid lineage. So it is important to kind of we tend to treat that as a tertiary marker and pair it with a brighter fluorochrome. The other thing too I want to mention is that because this goes to the process of fix and perm, it can be a bit harsher. So we try not to pair those with tandem dye. So I hope that's answered the questions.
Great, thank you. All right, I have another question here for you. Why should BSV be added to single color comp controls?
Another very good question. So yes, you should add it to the single control because it's important to treat your compensation control exactly the same way as you are treating your fully stained sample. And this is the very sort of standard practice for like CAP or any lab. They just want you to treat the control exactly the same way you treat with the sample.
Wonderful. Well, thank you so much, Kim. All right. We're getting some other questions in here. All right. Thank you for your patience. Okay, I have this next question here for you. This question asks, is BSB recommended to be added even if you're only using one BB dye? Also, have we documented any breakdown of some of the tandem BB dyes?
So it's yes, there are some breakdown, so first to answer that question, so the BV dye, if it's a tandem dye, then you treat it the same way. There's always a possibility of tandem dye breakdown, so you also need to be careful with that. Treat that, protect it from light, protect it from high temperatures. When you take it out, use it and put it right back into the fish. So then also the BSB only recommend if you have only 2 or more BV dyes used together. If you have only one, you don't need to add BSB.
Wonderful, thank you. Glad we got to answer that question here. All right. Great. And I think that was another question here. Perfect. All right, next question asks, with the BD Brilliant Buffer, would it be used only at the staining step?
Yes, so we use that before you add in the antibody.
Perfect. Great. Well, thank you so much, Kim. That will conclude our session for today. So I want to thank you again for your time and your important research. We would also like to thank Labroots and our sponsor, BD Biosciences, for underwriting today's educational webcast. Before we go, I'd like to thank the audience for joining us today and for their interesting questions. Questions we did not have time for today, and those submitted during the on-demand period, will be addressed by our speaker via the contact information you provided at the time of registration. This webcast can be viewed on demand, and Labroots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. So until next time, take care, everyone. Goodbye.